study both sexes to understand how all of us in the room work. I am not even going to start mentioning all of the awards, the International Society's leadership, journals, editor-in-chief, and what have you, study sections. It's just going to go on forever, and it'll be really boring because who you really want to hear is Professor Becker. So without any further ado, Jill, I am thrilled to have you here.
Thank you. And thank you so much for inviting me to be part of this fabulous symposium. I've really enjoyed my visit to UC Irvine. It's been a long time since I've been here, and I'm amazed at all of the growth and the amazing investigators who are here. I want to recommend to you, if you are doing work on sex differences, do I have a pointer? Please consider submitting your articles to the, Biology of Sex Differences journal that I am editor-in-chief of. And I also recommend the Organization for the Study of Sex Differences, which will be held in May in Calgary, Alberta. It's a nice meeting, and it's very collegial. So today, what I would like to talk to you about is the dynamics of sex differences in the brain, how the brain changes in response to hormonal and environmental signals during development, and how this impacts the reward system. I'm also going to talk to you about how estradiol modulates striatal dopamine release and stimulant drug-motivated behaviors. These are the mechanisms I've been studying for some time. And then I'll talk to you about how sex differences in addiction can be modulated by a number of factors, and in particular, social housing conditions as an animal model of social support, and estradiol receptor subtypes. And then we'll maybe talk about we're going to show you some of the new approaches that I'm using to study the neural mechanisms of dopamine release in freely moving rats. So just so that we're all on the same page, the brain develops from a bipotential organ. It can be masculinized or feminized, and the sex chromosomes determine whether you get testes or ovaries, and it's the testes producing androgens during critical periods of fetal development that masculinize the brain for the most part. There are also effects of sex chromosomes that can be measured and effects of the placenta, but the primary effect is due to androgens being produced by the testes and the absence of hormones being produced in females that result in sexual differentiation during the critical period. Do I have a pointer? Oh, okay. Okay, so, sorry. As I tried to point with my slide mover, it didn't work. So here we have the critical period during development, and then there's a second critical period at puberty, at which additional permanent changes to the brain emerge. So permanent changes in the brain start early in development. They are modified at puberty, and then activation of this brain, which has become different in males and females, is done by the hormones of the gonads, testosterone from males and estradiol and progesterone from females. Now, I want you to think about sex differences from a slightly different perspective than we usually think about them. I mean, usually we think that you have a dichotomous characterization and that there's no overlap. And for reproductive function or reproductive behavior, that's what you have. Males and females exhibit different behaviors, but not all sex differences are dichotomous. Some of them are an overlapping effect. You can measure the behavior on the same scale for males and females, and it's just that either males or females exhibit more of a behavior. That's what we usually for things like cognitive function and locomotor activity. That's what we usually think is going on. But if you start looking at your data more carefully, frequently you'll notice that you have population differences between males and females. And this can be that, for example, I'll show you that more females prefer cocaine over a tasty banana flavored pellets than males do. And this distribution, I believe, it can be affected by environmental events, stress during prenatal life, stress postnatally. And so your population of individuals who might be vulnerable to a particular phenotype can be affected by environmental effects. And then you have the complexity of how the brain is wired and how the brain responds to particular intracellular signals. And all bets are off here because the wiring is different in males and females in the reward system. And the CRH, for example, responds on the CR receptor 1 and receptor 2, both use different intracellular signaling mechanisms in males and females. So even though the hormones that are released are the same. So you have different mechanisms mediating the responses that we think are the same for males and females. So all of these things are involved. They're not individually, they're not mutually exclusive. So you can always, you always have to consider these different factors. Adrienne Beltz at the University of Michigan, along with Annalise Berry at my instigation, we wrote an article that I think helps people who aren't interested in studying sex differences put a handle on how they can begin to look at their data if they're trying to analyze data and figure out whether they've got to run more animals because they've got differences between males and females. This is in neuropsychopharmacology, and it just lets you put equations onto the descriptions that I've just given you and a way to think about your data. Now, I studied the reward system, but for the most part, I study dopamine and behaviors mediated by dopamine. So I want you to know that while I'll be focusing on VTA and substantia nigra dopamine systems, where the cell bodies are in the midbrain and projecting to dorsal striatum and nucleus accumbens, I'm completely aware that this circuitry is being modulated by prefrontal cortex and amygdala function, as well as the hippocampus. And all of this needs to be put together before we're really going to understand how these circuits are functioning together. I've been doing this a long time, and as you can tell by these references. And in many ways, so you should know that my study of the sex differences in striatal dopamine function was a control experiment gone awry. So I was studying sex differences in catecholamine control of the release of GnRH from the hypothalamus, and we decided to use the striatum as a control because it wouldn't have any sex differences, of course. But we didn't see anything in the hypothalamus and ended up with this interesting sex difference in the dorsal striatum, which is that amphetamine-stimulated dopamine release varies with the estrous cycle. It's modulated by estradiol. You take away your ovariectomized females, take away their estradiol, and then give it back to them, and you can reinstate potassium or amphetamine or electrical stimulation-induced dopamine release. And you went on to show over the years that estradiol applied directly to the dorsal striatum can modulate dopamine release, and you can put it directly into the dorsal striatum and enhance dopamine release. So in many ways, as you'll see, I have been redoing my dissertation experiments over and over again, but using more refined methodology. And estradiol is also rapidly downregulating D2 dopamine receptors. These effects of estradiol are rapid, which is to say, they're not the classical mechanism, which we think about intracellular functions that activate particular protein synthesis. These are rapid effects acting on membrane receptors for estradiol, and I'll tell you more about that in a minute. Okay. We've recently done in vivo microdialysis and showed that if you put estradiol either subcutaneously or directly into the dialysis probe compared to control, you get enhanced amphetamine-induced dopamine release in the dorsal striatum of ovariectomized female rats. So you can see this effect of estradiol in vivo and directly with the striatum. This is a funny phenomenon that happens when my slides are shown on a PC, and because this is not the figure that is supposed to be shown, it's a other figure from the same paper. I don't know how it happens. Anyway, the point is, you'll have to believe me because it's not quite as impressive from the horizontal slice, but estradiol 30 minutes later downregulates D2 dopamine receptors as shown by autoradiography in sections from female rats, but when you give estradiol to male rats, you don't see the same downregulation. So how is estradiol acting in the brain? We know that the nuclear receptors for estradiol are ER alpha and ER beta. And there are also there's a membrane receptor that for a long time was just called MER, and we weren't really sure what it is or what it was. And we now know that both ER alpha and ER beta can be processed after synthesis so that they'll then associate with caveolin. So they're palmitoylated, and then they associate with caveolin to then, in caveoli, which are little caves at the external membrane of a cell, they'll associate with metabotropic glutamate receptors and mediate the rapid effects of estradiol. And the specific mGluR depends on the brain region. So hippocampus and dorsal striatum are different, but they, so ER alpha and ER beta tend to prefer different mGluRs. So in the dorsal striatum, ER alpha associates with mGluR 5 and ER beta with mGluR 2, 3. And then recently we had to add GPER1, the G protein coupled estradiol receptor, which is a membrane receptor that rapidly mediates the response to estradiol because we found effects of it as well. Now, the brain isn't unique. Everywhere in the body, estradiol is acting at these particular receptors. Many of the effects of breast cancer and the amplification of the effects of estradiol in breast cancer are mediated by membrane effects of estradiol. So these are important receptors everywhere in the body. So here's a little cartoon to show you how this works. So estradiol combined to the GPER1 on the cell surface can bind to ER alpha or ER beta that will then go into the nucleus, but we're not really talking about those effects today. You can then also have ER alpha or ER beta associating with metabotropic glutamate receptors to mediate downstream cascades. So you have these multiple effects, and many of them happening in the same cells. So here's my cartoon of what I think is happening in the brain and in the dorsal striatum in particular. So you have the substantia nigra dopamine neurons that project to dorsal striatum, and I'm particularly interested in dorsal lateral striatum. That's where we see the main effects of estradiol. And in the dorsal lateral striatum, estradiol is acting on medium spiny GABAergic neurons to downregulate D2 dopamine receptors and downregulate GABA release. So estradiol acting at membrane estradiol receptors is decreasing GABA release. Okay, so here's the evidence that estradiol is acting by the metabotropic glutamate receptors in our system, the microdialysis. If you give, so estradiol alone to an ovariectomized female rat and then give her amphetamine, 30 minutes later, you get enhanced response compared to estradiol plus ICI 182780, which is an estradiol receptor antagonist at ER alpha and ER beta. If you give estradiol plus MPEP, which blocks metabotropic glutamate receptor 5, what you see is the effect of estradiol is prevented in the presence of the MPEP, which is the mGlor5 antagonist. We also used viral vector techniques to show that ER alpha is modulating GABA release, potassium-stimulated GABA release in dorsal striatum. So initially, this is an adeno-associated vector with ALP, which is just a housekeeping gene, and you can see that estradiol downregulates potassium-induced GABA increase in microdialysate. If you give animals who have or overexpressing ER alpha, what you see is you actually have a slight attenuation of the response to potassium in the overexpression of ER alpha. And when you give them estradiol, you can have a greater attenuation. So overall, there's more and a greater effect of estradiol in the presence of overexpression of ER alpha. And then Kristen Schultz, who was working with Bob Kennedy, but she did her PhD in my lab doing this, just also showed that if you look at the effect of estradiol with potassium-induced GABA release, MPEP prevents the effects of estradiol from happening compared to the saline. So again, with both GABA release and with dopamine release, estradiol is having rapid effects that are mediated by metabotropic glutamate receptors. Okay, so for a long time, I told everybody that this is the way it works in dorsal striatum, and so of course, that's the way it works in the nucleus accumbens. I don't believe that anymore. And it's more complicated. Everything's always more complicated, right? So what we showed is using ovariectomized females on this side and castrated males on this side, and we put a dialysis probe in the dorsal striatum on one side and in the nucleus accumbens on the other. So these are the same animals getting the same drugs. So we have ovariectomized females getting oil, and the dorsal striatum you get after 30 minutes after estradiol, you give, in this case, we're giving 10 mg per kg cocaine. You have this enhancement of dopamine in the dialysate, and you don't see an effect of estradiol in males, castrated males, 30 minutes later. Interestingly, though, in the nucleus accumbens, and our probes were in the core shell border, so we call it the shore of the nucleus accumbens. And what you see basically is that there's no effect of estradiol, but there was a significant sex difference in that females had significantly lower cocaine-induced dopamine increases in dialysate, which I think is important for mechanisms of addiction. Now, at that time, Katie Yost was in the lab, and we started transitioning to fast scan cyclic voltammetry because it's more rapid. And what Katie did was put fast scan probes into the nucleus accumbens because she was convinced that there was going to be an effect in the nucleus accumbens. It's just that we hadn't been specific enough. And so she gave ovariectomized and castrated animals. She looked at the stimulated dopamine release, then gave them estradiol or the ER alpha agonist PPT or the ER beta agonist DPN, looked at the stimulated response, gave them cocaine, and looked at the response again. And what she measured was the increase in dopamine, which is the maximum dopamine. We also looked at the reuptake, but I'll not tell you about that today. So what Katie found is that estradiol in females enhanced the stimulated release of EB after cocaine compared to the pre-cocaine response in females, but not in males. And if she gave PPT, the ER alpha agonist, there was no effect, but the ER beta agonist, DPN, did enhance or mimic the effects of estradiol to have enhanced dopamine release. no effect in males with either PPT or DPN. And at maximum currents, you get the biggest effect in females, but you also saw an effect of DPN even with fewer pulses of stimulation. Okay, so what I've shown you so far is that estradiol is rapidly enhancing stimulated dopamine release. And it's doing this by acting directly on the dorsal striatum. And I didn't show you data showing that it was acting directly on the nucleus accumbens, but Martin Kelly and other people have shown that as well. I also showed you that estradiol is rapidly downregulated dopamine receptors. So you're getting an upregulation in dopamine release, downregulation in the D2 receptors. And what I think is happening, if you think about the circuitry of the striatum, is you're shutting off the brake coming out of the striatum and turning on the go. So you're basically accelerating a system and preventing it from switching out of the current behavior. The lower dopamine release in the nucleus accumbens is going to make the females more readily transition from simply acquisition of drug taking to more habitual drug taking. And then the mechanisms are being mediated by the mGluR5 receptor. Okay, so how is this important for drug taking? As I told you, the dynamic brain during development is basically setting up the response, not only to estradiol, but the way the system is going to respond to drugs of abuse. Now, I know a lot of people come to me and say, why in the world would there be sex differences in motivation? You've got animals, everybody wants to eat, right? Everyone wants to make babies. So what's the difference? You know, why would there be a sex difference? And my biggest hunch about why this system is really organized differently is that it's related to maternal behavior, because female mammals need to be able to rapidly form attachments to to their offspring after parturition. And it's been shown in the sheep and in the rat that during parturition, there's an increase in dopamine release along with the increase in oxytocin, and that those two things are necessary for the formation of the mother-infant bond. So those two things are also happening during attachment to drugs of abuse. And so I believe that the neural system is designed to enhance this system in females compared to males. And the neural systems mediating reproductive behavior and reproductive function are different. And there are also sex differences in energy intake in the system that modulates that. I'm not going to talk about the latter two right now. If you look at humans and what is going on in terms of drug abuse, when I first started writing grants asking for people to give me money to study sex differences in addiction, and I had the audacity to propose that the females were going to be more vulnerable, would take more drugs. People said, well, that's ridiculous because everyone knows more men are addicts than women, and so it can't be that way. And at the time, it was about 80% men, 20% women. Right now, in the last 25 years, the difference has gone away in the high schools. It's now 50-50 equal opportunity for all drugs of abuse. Psychostimulants, girls are slightly higher. THC, boys are slightly higher, but overall, both boys and girls are taking drugs of abuse in high school, and the difference in the adult population is about 60-40, 60% men, 40% women. Women escalate drug use more rapidly than do men, and you can see this with cocaine and alcohol, as well as other drugs of abuse like heroin. Females present for treatment consuming more than men, even though their time of use is shorter. And their reasons for initiating drug use are different. Women report self-medicating for depression, and men report engaging in risky behaviors and wanting to be part of the group. So all of these things play into how we should be thinking about addiction treatment as well as the underlying mechanisms. So one of the first studies we did was just show that if you take ovariectomized females and you give half of them estradiol 30 minutes before self-administration. They get put into a little operant conditioning box. They can nose poke and get 1 injection of cocaine. And when you do that, 30 minutes after estradiol, what you see is that females getting hormone treatment take significantly more cocaine and learn to acquire cocaine taking more rapidly than ovariectomized females who just get oil, sham or intact males who are just getting oil, and castrated males who are just getting oil. That's a low dose of cocaine, 0.3 mg per kg per infusion over 5 consecutive days. They get 2 days off, and then we start over with a slightly higher dose. And what you see is that at 0.4 mg per kg per infusion, both ovariectomized females and females with estradiol on board have acquired cocaine self-administration compared to both of the male groups which have not. And that indicates that there are underlying organizational differences of hormones that result in enhanced acquisition of cocaine in females compared to males. At the highest dose, everybody takes cocaine, and but the females are still taking more if you measure the amount of cocaine that's being taken. Now, we've been trying to develop various paradigms, but let us ask questions that will make it more, like humans. Well, it can't really be, because they're rats and they live in cages. But, if I was a rat and I was put into this operant conditioning unit and I could nose poke for cocaine, and the rest of the time I'm in my colony room doing nothing, I think I'd probably nose poke for cocaine, right? So we thought it would be good to give them a choice. And so in this case, they had the choice of a banana flavored pellet or cocaine. And it's the kind of thing where they get 1 session with cocaine, 1 session off, 1 session with pellets, and that's randomized over the times. And then they get to choose at the end of the day for 30 minutes. And during the second week of training, when they're nose poking quite nicely during the no choice, sessions for both, what you see is that during the choice session, both females and males want pellets. So the pellets, are the most reinforcing to begin with, and the cocaine is really not being chosen. But 7 weeks later, what you see is the females, that some animals choose cocaine, and some animals choose pellets. And what we see is that once they go over to choosing cocaine, they don't go back to the pellets. And you might notice that it's also true that more females choose cocaine. So if you look at that over time, what you see is that females choose cocaine more rapidly, and about 50% of the females choose cocaine, but only about 20% of the males. And we've replicated that a number of times. So there is a population difference in animals who choose cocaine over pellets. If we look at some animals, even though they learn to nose poke for cocaine, really don't take much. And we call those abstainers. These are the pellet preferring animals. And some of the pellet preferring animals take just as much cocaine as the cocaine preferring animals. It's just when it comes to the choice session, they choose the pellet. And what we did was measure nucleus accumbens dopamine in animals at week 2, and then again at week 7. And what you'll see here is that there's no difference at week 2 to week 7 in the abstaining animals or the pellet preferring animals, but for the cocaine preferring animals at week 7, there's a significant decrease in cocaine-induced dopamine and dialysate. And this was true for both females and for males. So what we see is basically what Nora Valco has reported in humans with her PET imaging data, a decrease in stimulated dopamine associated with preference for cocaine. Okay, so which estradiol receptors are mediating the effects of motivation for cocaine and other drugs of abuse? We've talked about ER alpha and ER beta, but what about GPER1. Well, I hadn't even considered GPER1 until Jackie Quigley did this experiment in my lab for her dissertation. And she was looking at conditioned place preference in males and females. And our idea was if we blocked estradiol receptors using ICI 182780, that we would find that that would prevent conditioned place preference in females. So we thought it was a pretty simple experiment. But at the time, we hadn't really had our eyes open to the fact that this drug is also a GPER1 agonist. So to Jackie's surprise, what she found is that ICI blocked conditioned place preference for 10 milligrams per kilogram cocaine in males and had no effect whatsoever in females. Now, Jackie was a good student, so she went into the literature, found that ICI was also a GPER1 agonist, and then said, can I do the experiment to look at if I give a GPER1 agonist? And so that's what she did. And what she found is that G1, the GPER1 agonist, blocks the effects of cocaine to form a conditioned place preference in males, but it had no effect in females. She also showed that G15, the antagonist, enhances preference for a dose of cocaine that males do not usually form a conditioned place preference for. We did this in females as well, but they all already formed a preference for cocaine at 5 mg per kg, so it didn't do anything. So what she showed is that by inhibiting GPER1, you enhance preference for cocaine, and by activating GPER1, you get a decreased preference for cocaine on a conditioned place preference test. Jackie then went on to look at cocaine self-administration. And so what the animals did was they learned how to self-administer on a progressive ratio schedule, where that means for each additional treatment of the drug, they have to work harder for the next one, until finally they say, enough, I'm not working anymore, that's my breaking point. And so we're looking at what happens with a progressive ratio schedule. Then they got either G1 or cholesterol, and we looked at responding for the next 2 weeks. They were then withdrawn, and we looked at reinstatement. And so what you see is that GPER1, so during baseline, females had higher breaking points than males on average. And then when we gave G1, females actually increased their breaking points over controls, while the G1 did nothing for self-administration for the males. So obviously, conditioned place preference and self-administration are tapping into 2 different neural systems and neural mechanisms. When it came to reinstatement, G1 also enhanced reinstatement of responding for cocaine-induced reinstatement, and half of the animals had gotten G1 before and half cholesterol before. The X's were the cholesterol and pluses were the G1. So there was no effect of prior treatment. So now I'm really excited to share with you the stuff I'm doing now that has gotten me more excited than anything. We're working with Cynthia Chestick in biomedical engineering, and she has developed these carbon fiber array electrodes that allow us to look at 16 channels simultaneously using fast scan cyclic voltammetry chronically in freely moving animals. So what we do is we'll put our recording electrode in the accumbens or dorsal striatum, and there will be a stimulating electrode in the VTA or the medial forebrain bundle. So animals get their electrodes, we record, and then we get these little color plots where the green blip is dopamine. What's really cool is at the end of the experiment, the electrode stays in place, we decalcify the skull, and then we, for the dorsal striatum, we're staining with mu opiate receptor, tyrosine hydroxylase, and NuN, and in the nucleus accumbens, we're staining with calbindin for core, and Nguyen, so that we can tell which subregions of each brain region we are. And Noah Leonardo, who's a new graduate student with me, she has done a lot of work to make this beautiful. I don't know if this will work. So, okay, this is a 3D image showing that there's very little damage. I don't know if I can. No, it's really pretty. We'll go on. Okay, so this is just to show that over a 4-week period of time, this is just 4 of the 16 channels of data that we get every 15 milliseconds. So we have a very dynamic response and a lot of data. But the response, you can see, week 1, you have less of a response than you do on subsequent weeks. There's a bit of conditioning of the electrode that goes on over time. But it's a stable response, and we have, since we have 16 channels, this is just a typical one, this wasn't beautiful or anything. We were able to follow dopamine over up to 4 weeks. So one of the questions we've been asking with this method is, what does social housing do in male and female rats? And we ask this because if you socially house, just give animals a friend in their home cage, that's all, or not. What we see is that individual females who don't have a friend in the home cage take more cocaine, and they'll work harder for cocaine than socially housed females. But we don't see an effect of social housing in males. Yvette Gonzalez is working on this, and she's doing fabulous work. She's also quantifying the social behaviors that the animals are engaging in their home cage. But what she's shown is that over 3 weeks of time, social housing affects stimulated dopamine release in the nucleus accumbens of female rats, but not males, and particularly in the shell, where the social housing enhances the dopamine release of the females, but social housing is not enhancing the response in males in either the core or the shell. There's some effect of social housing in the core as well. Then the other thing we've been doing is looking at, of course, in dorsal lateral striatum to look at dopamine release. And first, over the 4 weeks of recording, Chris Turner has shown that there is enhanced stimulated dopamine release by stimulating the medial forebrain bundle in both males and females, and this is pretty reliable across time. And then we're doing the experiment of my dissertation experiment one more time. And this is basically looking at the effects of estradiol and dorsolateral striatum, but measuring with fast-scan cyclic voltammetry. So this is a little confusing. These are the control animals, and at each, this is before an injection, and then the controls just get oil, oil, oil. Then this is G1, so this is control injection, and then they're getting 5 micrograms of G1 at each of the 3 injections, so the cumulative dose is 15 mg per kg G1, and then they get 1 injection of estradiol before injection 1, and then just oil injections. So this is the acute response to estradiol, which was not present in the males. But in the females, what you see is that we are seeing an effect of G1 in dorsolateral striatum and replicating the effect of estradiol to enhance stimulated response after estradiol. So we're excited to be using this. There's so much data. We've got like 3 papers we're trying to get out because we're finally getting the analysis done. But this is going to be Chris Turner's dissertation. He's almost done. And he's fabulous, by the way. Okay, so I've told you that there are different types of sex differences. And as you look at the story that I've been telling you, what you've seen is that there are quantitative sex differences in this model. You've got the effects of estradiol. You've got the initial response to cocaine or amphetamine, which is greater in females than in males. But you also have population differences in the acquisition of or preference for cocaine over pellets. And we have differences in the mechanisms in the GPR1 mediated effects on the reward system. So when we're thinking about addiction, all of these different types of sex differences apply. So, the brain is dynamic in many ways. As Emily pointed out earlier, hormonal influences are just one piece of it. It's also dynamic in that developmental influences are going to be interacting with the hormonal influences to result in sex differences in drug taking in males and females. GPER1 looks to be an important player in attenuating the preference for cocaine in males, but enhancing motivation for cocaine in females. So we need to try to understand the neural circuitry that's important for condition place preference versus self-administration of cocaine, because clearly different systems are working in these cases. And estradiol is acting at ER beta in the nucleus accumbens shell, ER alpha in the dorsal striatum, and GPR1 is important in the enhancing dopamine release in the dorsolateral striatum as well. So as Emily got on her soapbox at the end of her saying studying females are important, I hope you agree that and understand that this is really important to our understanding of the brain. If you're going to understand the brain at all, you need to know how it works in all of the individuals that are part of your population, because sex differences are really fundamental to how the brain works. And the more we can understand sex differences and similarities, the better treatment we can provide to people for all different kinds of mental disorders. Okay, Cynthia Chestik is the brilliant biomedical engineer along with Paresh Patel. Diamond Kai is the neuroanatomist who's been helping us get those beautiful images. And Chris Turner, Kristen Lee, Yvette Gonzalez, Noah Leonardo, Brandon Luma have really been doing the FSCV work and have got getting it going. And this is my lab, so thank you. I'm happy to take questions.
Okay, questions to Dr. Becker. Go ahead. Is there a microphone there? Good.
Mr. Ivani, good to see you. Great talk. I have two questions. One, kind of, you knew it probably going to bring up the influence of sex chromosome component in reward systems. And now there's a recently published preprint for protein type in rats. So what are your thoughts about this? How much do you think sex chromosome component influences sex differences versus differences in life?
So I think this sex hormone complement is important, especially in the function of the TH neurons, because as you've shown that there is SRY in the TH neurons. And we've also seen in work from Jane Taylor that there's effects of sex chromosome complement on motivation for reward. So I think it's important. And with the 4 core genotype rats that Art is about to publish, I've seen the preprint too. I'm excited to be able to answer and look at those questions in rats.
Very quick. Second order questions is about the interaction and sex as a biological variable, which, if you could comment as a leader in the field of sex differences and in OSSD, is it effective? Is it, there's clearly there's a mandate to write something. Is it become perfunctory? Because the third point is, NIH has never provided more money to double the amount of whatever sample that you use. So does it actually create the danger to actually reduce poverty?
So I would argue that because you can't do as many experiments using only male animals, that it's not that you're not learning more when you also include females. You're still doing as many experiments, you just simply have more groups, and you're learning something fundamental about how the brain works. And what I've I've seen talking to people around the country is that there are some really interesting findings out there. And as far as I'm concerned, I'll say this out loud, this is the low-hanging fruit in neuroscience. Figuring out what's going on in females. Come on, you guys. This is not brain science. This is really, this is rocket science. This is just brain science. Understand what's going on in females, and you'll actually learn a lot more than we know right now.
I'd like to simply mention that Dr. Eric Villain, welcome Eric, as the new director of the UCI CTSA. So I think that is really something that your question and probably many others. Personally, I would like to violently agree with Dr. Becker. I think that we find this in Conte Center, at least, both in humans and in experimental animals, that we have such strong organizational effects, namely pre-pubertal, pre-hormonal, if you will, that we must really study both sexes to understand the brain. Welcome, Eric. Questions to Dr. Becker?
I mean, and if nothing else, people simply have to report their numbers for males and females in a paper, so that those of us who are really interested in the sex differences can begin to look for the patterns that are there. And with data repositories becoming mandatory, we can, those of us who are into really understanding sex differences could begin to pull those out. And those of you who don't want to do that, at least you'll have reported the numbers so that we can begin to figure out what's different and how the different neural systems are working. But it's not just quantity, it's circuitry. The circuitry is different. It's a fundamental difference in how the brain is controlled, I think because of the hypothalamic organization, which by necessity of reproduction is different, and pregnancy, which means you have to have different immune systems, different thermoregulatory systems, different sleep systems. I mean, all of it is designed to make both animals, males and females, work in the same environment, right? And if you've got different core systems to hook up to in order to make the animal go out for food and eat in the appropriate amount and thermoregulate, the cognitive systems that control those systems and interact with those systems also have to be different.
Questions to Dr. Becker? I have one question that actually relates to the data. It's not a big question, it's a tiny question. When you looked at the effect of GPR1 and estradiol, you found, G1 antagonist, you found that both increased dopamine release in the umpteenth version of your thesis experiment, the most recent one, which is very exciting.
Right.
What happens if you give them together? Because I think that if they are working through the same pathway, one would perhaps predict that they will be occluded. I mean, they won't be additive. Whereas if they're not working quite to the same pathway, you would have an additive effect. So have you done that, and what did you find?
We haven't done that yet, but that's definitely something that has to be done. We have to try to antagonize the effects of estradiol. As you know, estradiol has a bell-shaped curve, which is to say, if you work within physiological concentrations of estradiol, you'll see this enhancement. If you go too high, it actually is inhibitory. So I would expect if we tried to give both estradiol and GPER1 at the same time, we'd actually see a reduction. And if you didn't know about the bell-shaped dose-response curve, it would confuse you. But keep that in mind.
Other questions? If not, let's get a major.
There's one more question.
Oops. Go
I
ahead.
have one from online. Have you looked at how estrogen receptor signaling interacts with glucocorticoid signaling? Considering sex differences in HPA axis regulation, it seems plausible that there are differential interactions between sex and stress hormones, depending on their relative levels and receptor expression.
So that's a great question. I haven't done that, but we know that there are even sex differences in the glucocorticoid receptors functioning, CRH as well as GR and MR. So it's bound to be an important component of it. Yeah, I encourage anyone who's set up to do that to investigate that, and I'm happy. Actually, Shelley Flagel has a graduate student for whom that question is key, and that's going to be part of her dissertation, and we'll be collaborating doing FSCV on that. So hold on, I take it back, we will be looking into that.
All right, let's hear a major shout to Dr. Becker, and thank you everybody for this.